Hello everyone, welcome to the next lecture in our physiology video lecture series. This particular lecture is titled Respiration Part 1. In this particular lecture, we will look into pulmonary ventilation, the structure and function of the lungs, and the various aspects of the respiratory system and how exactly the respiratory system functions. So let us start. The first aspect we will look into is pulmonary ventilation. The goal of respiration is obviously to provide oxygen to the tissues and remove the end product, carbon dioxide. This is achieved by the following functions. The first one is pulmonary ventilation itself, which means the inflow and outflow of air between the atmosphere and the lung alveoli. The next function is diffusion of oxygen and carbon dioxide between the alveoli and the blood. Third is the transport of oxygen and carbon dioxide in the blood and body fluids to and from body's tissue cells. So obviously, the various aspects of respiration can be divided into three major parts. First is the inflow of fresh air into our lungs, which falls under pulmonary ventilation, and the expiration or outflow of the same. Next is the diffusion of oxygen and carbon dioxide across the pulmonary capillaries and into the blood in case of oxygen and out of the blood in case of carbon dioxide. And final part of the respiration is the transport of oxygen and carbon dioxide in the blood and body fluids to and from body's tissue cells once they have been exchanged at the level of the lung alveoli. The blood cells try and take the oxygen from the lungs to different parts of the body and recover carbon dioxide from different parts of the tissue, different parts of the bodies and tissues back to the lung itself. So transport of oxygen carbon dioxide is the third arm of respiratory system. Next, we also will look into regulation of ventilation and other facets of respiration. How exactly the rate at which expiration and inspiration occurs are controlled is what we will look into as well. But first, we'll look into mechanics of pulmonary ventilation itself. The lungs can be expanded and contracted in two major ways. The first is by downward and upward movement of the diaphragm to lengthen or shorten the chest cavity, or by elevation and depression of the ribs to increase and decrease the anterior posterior diameter of the chest cavity. So as you can see in the diagram right here, we have shown different muscles and which help in expiration and inspiration via the lungs. So in case of expiration, we see that the abdominal muscles contract and shorten the lungs, which are present right here. And this is what happens in case of expiration. But instead, in case of inspiration, the diaphragm contracts, which is the muscle layer just beneath our lungs, and this causes the lungs to expand and thus pull air into it. We'll look into the mechanics of pulmonary ventilation in the next slide. So, normal quiet breathing is accomplished entirely by the first method, that is by the movement of the diaphragm itself. During inspiration, the contraction of the diaphragm pulls the lower surfaces of the lungs downwards. Then during expiration, the diaphragm simply relaxes and the elastic recoil of the lungs, chest wall and abdominal structures compresses the lungs and expels the air. During heavy breathing, however, the elastic forces are not powerful enough to cause necessary rapid expiration. So that extra force achieved mainly by contraction of the abdominal muscles, which pushes the abdominal contents upward against the bottom of the diaphragm, thereby compressing the lungs. Thus, in case of normal breathing, the diaphragm itself can handle the expiration and inspiration process via its own contraction and relaxation. But during heavy breathing, when the body is under strain and the lungs have to function at a higher level, the diaphragm itself cannot handle the rate at which the expiration and inspiration occurs. In that case, the abdominal muscles also generate extra force by pushing against the lungs and the diaphragm, and thereby help in increasing the rate and force of expiration and inspiration. The second method for expanding the lungs is to raise the rib cage. So in the first method itself, we will see a increase in the lungs. We will see an increase in the lung cavity along this axis. But in case when the ribs themselves expand, we will see an increased AP diameter. That is an increase in the anterior posterior diameter. So in one case, we will see an expansion along the anterior posterior axis, and in another case, we will see an expansion along this axis. Thus, the second method for expanding the lungs is to raise the rib cage. This expands the lungs because in the natural resting position, the ribs slant downwards, as shown in the left side of the figure, thus allowing the sternum to fall back towards the vertebral column. But when the ribs cage is elevated, the ribs project almost directly forward so that the sternum also moves forward away from the spine, making the anterior or posterior thickness of the chest about 20% greater during maximum inspiration than during expiration. Therefore, all the muscles that elevate the chest cage are classified as muscles of inspiration, and those muscles that depress the chest cage are classified as muscles of expiration. Thus, this is how the mechanism of pulmonary ventilation occurs. The most important muscles that are not shown in the diagram that raise the rib cage are the external intercostals. But there are other muscles which also help in the same, which include the sternocleidomastoid muscles, which lift upward the sternum, and the anterior serratii, which lift many of the ribs. Also helping in this function, that is raising the rib cage, the scalene muscles also play a major role in it, which lift the first two ribs only. The muscles that pull the rib cage downwards, that is the muscles which cause expiration, are mainly the abdominal recti, which have the powerful effect of pulling downwards on the lower ribs at the same time that they and other abdominal muscles also compress the abdominal contents upward against the diaphragm, and the internal intercostals also play a major role in expiration. Next, we move on to movement of air in and out of lungs and various pressures that act on the lungs themselves. The lung is essentially an elastic structure that collapses like a balloon and expels all its air through the trachea whenever there is no force to keep it inflated. So we can imagine a balloon itself when there is no force that allows the balloon to keep itself inflated and the opening is kept open, then all the air from the balloon will go out and thus deflate the balloon. The same is the case with the lungs. Whenever there is no force that is keeping it actively inflated, whenever the trachea is open, which is the windpipe, all the air that is present in the lung can just simply escape and thus deflate the lung completely. Also, we have to keep in mind that there are no attachments between the lung and the walls of the chest cage. Instead, the lung floats in the thoracic cavity, surrounded by a thin layer of pleural fluid that lubricates movement of the lungs within the cavity. Continuous suction of excess fluid into the lymphatic channels maintains a slight suction between the visceral surface of the lung pleura and the parietal pleural surface of the thoracic cavity. Now we'll look into the various pressures that act on the lung. The first pressure we'll look into is known as the pleural pressure. Pleural pressure is the pressure of the fluid in the thin space between the lung pleura and the chest wall pleura, which ranges from -5 to -7.5 centimeters of water. This pressure is required to hold the lungs open to the resting level while expansion respectively. Thus, this is the pressure that is essential in keeping the lungs inflated and does not allow them to deflate without any prior notice. Next, we look into alveolar pressure. Alveolar pressure is the pressure of the air inside the lung alveoli. When the glottis is open and no air is flowing in and out of the lungs, the pressure in all parts of the respiratory tree, all the way to the alveoli, are equal to atmospheric pressure, which is considered to be a zero reference pressure in the airways. That is 0 centimeters of water pressure. Next, we look into the transpulmonary pressure. Finally, if we focus on the diagram itself, we'll note that the figure, the difference between the alveolar pressure and the pleural pressure is the transpulmonary pressure. It is the pressure difference that between the alveoli and the outer surface of the lungs, and it is a measure of the elastic forces that in the lung that tend to collapse the lung in each instant of respiration, called the recoil pressure. So this pressure itself acts in, or it tries to deflate the lung, and is equal to the difference between the alveolar pressure and the pleural pressure. And in this particular plot, we have plotted the alveolar pressure that it acts in the lung, as well as the pleural pressure and the transpulmonary pressure. And in the upper part, we can see the lung volume as it increases during every cycle of expiration and inspiration. Next, we look into the compliance of lungs. The extent to which the lungs will expand for each unit increase in transpulmonary pressure is called the lung compliance. The total compliance of both lungs together in normal adult human being averages about 200 milliliters of air per centimeter of water transpulmonary pressure. So here we have a compliance diagram of the lungs, which consists of an expiration part and an inspiration part. The two curves that are given here are called the inspiratory compliance curve and the expiratory compliance curve. And the entire diagram is called the compliance diagram of the lungs. The characteristics of the compliance diagrams are determined by the elastic forces of two kinds that act on the lungs. These include the elastic forces of the lung tissue itself and elastic forces caused by surface tension of the fluid that lines the inside of the alveoli and other lung air spaces. We'll look into the surface tension and how it acts and helps in the compliance of lungs in the next slide. So we are looking into surfactant, surface tension, and collapse of alveoli. As we know, the alveoli are obviously wet from the inside, that is, they have a liquid surface or an inner water surface. Inside the alveoli, the inner water surface is trying to contract due to surface tension. This forces the air out of the alveoli through the bronchi and causes the alveoli to try to collapse. The net effect is to cause an elastic contractile force of the entire lung known as the surface tension elastic force. So you can consider the water droplet. In case of a water droplets always form a spherical structure. The reason behind that is that on the surface of the water droplet, there is an air-water interface. This air-water interface forces the water on the water molecules on the surface to describe a surface tension. That is the force that causes the water to make sure that the air-water interface is as minimum as possible. This force then contracts the whole water droplet such that it forms a spherical surface, as spheres have the least area in the particular volume that they can contain, and thus this minimizes the surface area and also the surface tension of the water drop. Similarly, in this case, the alveoli, which have an inner water surface, try to collapse in on themselves in order to reduce the surface area due to surface tension. And this in this particular context is known as the surface tension elastic force that acts on the lungs. Now, in order to deflect the surface tension elastic force, various parts of the lung alveoli secrete a special surfactant. Surfactant is a surface active agent in water, which means that it greatly reduces the surface tension of water. It is secreted by special surfactant secreting epithelial cells called type 2 alveolar epithelial cells, which constitute about 10% of the surface area of the alveoli. Surfactant is a complex mixture of several phospholipids, proteins, and ions. The phospholipid dipalmitoyl phosphatidylcholine, along with several less important phospholipids, is responsible for reducing the surface tension. The mechanism via which phospholipids reduce the surface tension is that they do not dissolve uniformly in the fluid lining of the alveolar surface. Instead, part of the molecule dissolves while the remainder spreads over the surface of the water in the alveoli. This surface has 1/12 to 1/2 of the surface tension of a pure water surface. In quantitative terms, the surface tension of different water fluids is approximately the following. Pure water has a surface tension of about 72 dynes per centimeter, whereas which is the normal fluid lining. This was pure water, which the surface tension with respect to pure water. Surface tension in case of normal fluids lining the alveoli but without any surfactant is about 50 dynes per centimeter. But normal fluids lining alveoli with normal amounts of surfactant is between 5 to 30 dynes per centimeter. That is a huge decrease in surface tension. And now we'll look into how exactly the surfactant and surface tension affect the compliance of the lungs. The elastic forces of the lung tissue are determined mainly by elastin and collagen fibers interwoven among the lung parenchyma. The elastic forces caused by surface tension are much more complex. The significance of surface tension is shown in the following figure, which compares the compliance of the lungs when filled with saline solution and when filled with air. The right hand side is obviously the air filled lung and the left hand side is the saline filled lung. When the lungs are filled with air, there is an interface between alveolar fluid and the air in the alveoli. Thus, we have a air-water interface that is present. In case of saline solution filled lungs, there is no air fluid interface present. Therefore, the surface tension is not present and only tissue elastic forces are operative in the saline solution filled lung. And thus we see in that case, the compliance of the lung has dropped dramatically. And thus, in the saline filled lungs, you can see that the compliance is dramatically increased. This is because there is no surface tension present to oppose the pleural pressure and subsequent lung volume change. This condition would have helped us enormously, but obviously, for practical application of the lungs, there needs to be a air-water interface present, and thus we have the normal condition, that is the right hand side, where there is a substantial amount of compliance that is present in the lungs. But In case of saline-filled lungs, where there is no air-water interface, the compliance is dramatically increased. That is, controlling the volume. Controlling the volume of the lung is very easy. Next, we look into pressure in occluded alveoli caused by surface tension. If the air passages leading from the alveoli of the lungs are blocked, the surface tension in the alveoli tends to collapse the alveoli. This creates positive pressure in the alveoli, attempting to push the air out. The amount of pressure generated in this way in an alveolus can be calculated from the following formula, which is pressure equal to 2 times the surface tension divided by the radius of the alveolus. For the average sized alveolus with a radius of about 200 micrometers and lined with normal surfactant, this calculates to be about 4 centimeters of water pressure, which is around 3 millimeters of mercury. If the alveoli were lined with pure water without any surfactant at all, the pressure would calculate to about 18 centimeters of water, that is 4.5 times greater than the normal amount. Thus, we can see that the alveolar surfactant is very helpful in reducing the alveolar surface tension, and therefore also reduces the effort required by the respiratory muscles to expand the lungs. Next, we will look into the effect of alveolar radius on the pressure caused by surface tension. Following the previous formula, that is this one, the smaller the alveolus, the greater the alveolar pressure caused by surface tension. Thus, when the alveoli have half the normal radius, that is say 50 instead of 100 micrometers, the pressures noticed earlier are double. Thus, this is significant in case of small premature babies, many of whom have alveoli with radii less than a quarter that of adult person. Further, surfactant does not normally begin to be secreted into the alveoli until between the 6th and the 7th month of gestation. Therefore, many premature babies have little or no surfactant in the alveoli when they are born, and their lungs have an extreme tendency to collapse, sometimes as great as 6 to 8 times that in a normal adult person. This causes the condition called respiratory distress syndrome of the newborn. And this is a fatal disease, and if not treated with strong measures, especially properly applied continuous positive pressure breathing, which is one of the only ways in which this respiratory distress syndrome of the newborn can be stopped and the newborn can be helped to survive. Next, we will look into say some parameters related to the lungs and the volume of air that can be present in the lungs. The first thing we will look into is known as pulmonary volumes. The first volume that is a characteristic parameter of a lung is the tidal volume. The tidal volume is the volume of air inspired or expired with each normal breath. It amounts to about 500 milliliters in an adult male. So this particular, in this particular diagram, we have plotted the lung volume versus time, and the tidal volume is only a small volume that can be withheld in the lungs, and it is the normal air that is inspired or expired with each breath. Next is the inspiratory reserve volume. The inspiratory reserve volume is the extra volume of the air that can be inspired over and above the normal tidal volume when a person inspires with full force. It is usually equal to about 3000 milliliters, which is shown right here. This large peak, which denotes the entire air present in the lungs that the person can forcefully expire. forcefully inspire. Next is the expiratory reserve. The expiratory reserve volume is the maximum extra volume of air that can be expired by forceful expiration after the end of a normal tidal expiration. This normally amounts to about 1100 milliliters. And obviously, we can see that the expiratory reserve volume is on the opposite side to the inspiratory one, because in one case we are allowing more air to come into the lungs, and in another case we are expiring air out of the lungs. The final pulmonary volume is the residual volume. The residual volume is the volume of air remaining in the lungs after the most forceful expiration. This volume averages about 1200 milliliters. And this yellow region is the residual volume in the lungs. Next, we will look into pulmonary capacities. The inspiratory capacity equals the tidal volume plus the inspiratory reserve volume. This is the amount of air which is about 3500 milliliters a person can breathe in, beginning at the normal expiratory level and distending the lungs to the maximum. So this is the inspiratory capacity is this particular peak right here, which involves 1/2 or 1 part of the tidal volume and the excess amount of air that a person can forcefully inspire. Next is the functional residual capacity, which equals the expiratory reserve volume plus the residual volume. This is the amount of air that remains in the lungs at the end of a normal expiration. So this is the functional residual capacity that is shown in the plot. Next, we look into the vital capacity, which equals the inspiratory reserve volume plus the tidal volume plus the expiratory reserve volume. This is the maximum amount of air a person can expel from the lungs after first filling the lungs to the maximum extent and then expiring to the maximum extent that they possibly can. The vital capacity is about 4600 milliliters. Next is the total lung capacity, which is the maximum volume to which the lungs can be expanded with the greatest possible effort, which is about 5800 milliliters. It is equal to the vital capacity plus the residual volume. And this is the total lung capacity which spans the entire part of the plot. All pulmonary volumes and capacities are about 20 to 25% less in women than in men, and they are greater in larger and athletic people than in small and asthenic people. So after looking into the pulmonary volumes and capacities, now we move into the respiratory passageways. The first respiratory passageway are the trachea, bronchi, and the bronchioles. One of the most important problems in all the respiratory passageways is to keep them open and allow easy passage of air to and from the alveoli. To keep the trachea from collapsing, multiple cartilage rings extend about 5/6 of the way around the trachea. In the walls of the bronchi, less extensive curved cartilage plates are present, which also maintain a reasonable amount of rigidity, yet allow sufficient motion of the lungs to attract and contract. These plates become progressively less extensive in the later generations of the bronchi as we progress along the lung respiratory tree, and are gone in the bronchioles, which usually have a diameter of less than 1.5 millimeters. The bronchioles are not prevented from collapsing by the rigidity of their walls. Instead, they are kept expanded mainly by the same transpulmonary pressures that expand the alveoli. Next, we look into the muscular wall of the bronchi and the bronchioles and its control. In all the areas of the trachea and bronchi that are not occupied by cartilage plates, the walls are composed mainly of smooth muscle. Also, the walls of the bronchioles are almost entirely smooth muscle, with the exception of most terminal bronchiole, which is known as the respiratory bronchiole, which is mainly pulmonary epithelium and underlying fibrous tissue plus a few smooth muscle fibers. Many obstructive diseases of the lungs result from narrowing of smaller bronchi and larger bronchioles, often because of excessive contraction of the smooth muscle itself, which in turn closes off the bronchioles and thus prevents the lung alveoli from being filled. Next, we look into the resistance to airflow in the bronchial tree. Under normal respiratory conditions, air flows through respiratory passages so easily that less than 1 centimeter of water pressure gradient from the alveoli to the atmosphere is sufficient to cause enough airflow for quiet breathing. The greatest amount of resistance to airflow occurs not in the minute air passages of the terminal bronchioles, but in some of the larger bronchioles and bronchi near the trachea. The reason for this high resistance is that there are relatively few of these larger bronchi in comparison to approximately 65,000 parallel terminal bronchioles, through each of which only a minute amount of air must pass. Thus, after looking into the various musculature as well as structure of the respiratory passageway, we now need to look into the regulation of the bronchiolar musculature. The bronchiolar musculature can be regulated by two major ways. One is the nervous control, and the next is the local secretory factors that control bronchiolar musculature. First, we will look into the nervous control. Obviously, the nervous control is again divided into sympathetic and parasympathetic parts. The sympathetic, which prepares the body for the flight or fight response, or the parasympathetic, which prepares the body for a resting response. So first we look into sympathetic dilation of the bronchioles. Direct control of the bronchioles by sympathetic nerve fibers is relatively weak because few of these fibers penetrate to the central portion of the lungs. However, the bronchial tree is very much exposed to norepinephrine and epinephrine released into the blood by sympathetic stimulation of the adrenal gland medulla. Thus, the nerves themselves do not directly innervate the lungs and the alveoli, but instead they stimulate the adrenal medulla to release norepinephrine and epinephrine, which in turn have a similar effect on the lungs. Next is the parasympathetic constriction of the bronchioles. A few parasympathetic nerve fibers derived from the vagus nerves penetrate the lung parenchyma. These nerves secrete acetylcholine and, when activated, cause mild to moderate constriction of the bronchioles. When a disease process such as asthma has already caused some bronchial constriction, superimposed parasympathetic nervous stimulation often worsens the situation. When this occurs, administration of drugs which block the effects of acetylcholine, including atropine, can sometimes relax the respiratory passageways enough to relieve the obstruction. This was all about the nervous control of the bronchiolar musculature. We now move on to the second type of regulation, which is via local secretory factors. Several substances formed in the lungs themselves are often quite active in causing the bronchial constriction. Two of the most important of these are histamine and slow reactive substance of anaphylaxis. Both of these are released in the lungs tissues by mast cells during allergic reactions, especially those caused by pollens in the air. Therefore, they play key roles in causing the airway obstruction that occurs in allergic asthma. This is especially true of slow reactive substance anaphylaxis. The same irritants that cause parasympathetic constrictive reflexes of the airways, which include smoke, dust, sulfur dioxide, and some of the acidic elements in smog, often directly on the lung tissues to initiate local non-nervous reactions that cause obstructive constriction of the airways. Next, we look into the respiratory passageways. This is a simplified schematic diagram of the various respiratory passageways that are present, which include the glottis, the larynx, which are the vocal cords, followed by the trachea or the windpipe, and finally, via the trachea, we enter into bronchioles, and then finally into the alveoli that are shown right here. And in the alveoli, we have the exchange of CO2 and oxygen the blood with the blood cells that are passing in the pulmonary capillary. The interface for the exchange is the pulmonary capillary membrane, via which only the gases, oxygen and CO2 can pass through. Next, we look into pulmonary circulation, pulmonary edema, and pleural fluid. First, we look into pulmonary vessels themselves. The pulmonary artery extends only 5 centimeters beyond the apex of the right ventricle and then divides into the right and the left main branches that supply blood to the two respective lungs. The pulmonary artery is thin with a wall thickness one-third that of the aorta. The pulmonary arterial branches are very short, and all the pulmonary arteries, even the smaller arteries and arterioles, have larger diameter than their counterpart in the systemic arteries. This, combined with the fact that the vessels are thin and distensible, gives the pulmonary arterial tree a large compliance, averaging almost 7 milliliters per millimeter of mercury, which is similar to that of the entire systemic arterial tree. This large compliance allows the pulmonary arteries to accommodate the stroke volume output of the right ventricle. The pulmonary veins, like the pulmonary arteries, are also short. They immediately empty their effluent blood into the left atrium to be pumped by the left heart through the systemic circulation. This was the physiologic anatomy of the pulmonary vessels themselves. Next, we will look into the bronchial vessels. Blood also flows to the lungs through small bronchial arteries that originate from systemic circulation, amounting to about 1 to 2% of the total cardiac output. This bronchial arterial blood is oxygenated, in contrast to the partially deoxygenated blood in the pulmonary arteries. It supplies supporting tissues of the lungs, including the connective tissue, septa, and the large and small bronchi. Thus, the blood traveling through the bronchial vessels is the supplies oxygen to the muscles of the lungs themselves. So, even though the lung helps in the transfer of this large amount of oxygen throughout the whole body, the lung does not. The lung and the tissues present in the lung do not use the oxygen that is being transferred via the pulmonary artery into the heart. Instead, they depend on the bronchial vessels to try to transport oxygen to them. After this bronchial and arterial blood has passed through the supporting tissues, it empties into the pulmonary veins and enters the left atrium, rather than passing back to the right atrium. Therefore, the flow into the left atrium and the left ventricular output are about 1 to 2% greater than the right ventricular output. This is because obviously that the bronchial vessels contribute directly to the to the left ventricular output by transferring a small amount of oxygenated blood, which travels via the lung tissue directly into the left ventricle. Next, we look into the lymphatics. Lymph vessels are present in all supportive tissues of the lungs, beginning in the connective tissue spaces that surround the terminal bronchioles, coursing to the hilum of the lung, and hence mainly into the right thoracic lymph duct. Particulate matter entering the alveoli is partly removed by the way of these channels, and plasma protein leaking from the lung capillaries is also removed from the lung tissues, thereby helping to prevent pulmonary edema. So this was in this particular slide we have looked through pulmonary circulation, edema, and pleural fluid. We are looking specifically at the pulmonary vessels, bronchial vessels, and the lymphatic system present in the lungs. Next, we will move into pressures in the pulmonary system. The first is we'll look into the pressure pulse curve in the right ventricle. In this particular slide, we are only looking at blood pressures that are present in the various pulmonary vessels that are present in the lung. The pressure pulse curves of the right ventricle and the pulmonary artery are shown right here. The systolic pressure in the right ventricle of the normal human being averages about 25 millimeters of mercury, and the diastolic pressure averages about 0 to 1 millimeter mercury, which are values that are only 1/5 of those are for the left ventricle. The pressure in the pulmonary artery. During systole, the pressure in the pulmonary artery is essentially equal to the pressure in the right ventricle. After the pulmonary valve closes at the end of the systole, the ventricular pressure falls precipitously. Thereby, the pulmonary artery pressure falls more slowly as blood flows through the capillary in the lungs. The systolic pulmonary artery pressure averages about 25 millimeters of mercury in the normal human being. The diastolic pulmonary arterial pressure is about 8 millimeters mercury, and thus the mean falls about 15 millimeters mercury. And here we can see the in the pressure curve that the in the systolic part of the right ventricle, the pressure curve follows almost equally in case of right ventricular curve as well as the pulmonary artery curve. Thus, when the right ventricle is in the systolic phase, the pressure in the pulmonary artery and the right ventricle is almost equal. But in the diastolic phase, as the pressure in the right ventricle reduces dramatically, the pressure in the pulmonary artery does not, as the blood present in the pulmonary artery and the pulmonary arterial tree still contributes to a nominal pressure that is maintained and shown by the red line till the second systole is activated, and this now again the pulmonary artery curve follows the right ventricular. Next, we look into the pulmonary artery capillary pressure, pulmonary capillary pressure. The mean pulmonary capillary pressure is of 7 millimeters of mercury. This low capillary pressure is important for fluid exchange function of the pulmonary capillary. Finally, we will look through the left atrial and the pulmonary venous pressure. The mean pressure in the left atrium and the major pulmonary veins averages about 2 millimeters of mercury in the recumbent human being, which is the resting human being. And in this particular diagram, we can see that while in the pulmonary artery, there is a pulsatile nature of blood pressure, but while the blood reaches the pulmonary capillaries, the pulsatile nature has almost dropped out, and we get a linear decrease in blood pressure as the blood travels via the pulmonary capillaries to the left atrium. Next, we look into pulmonary capillary dynamics. The first thing we look into is the pulmonary capillary pressure. Isogravimetric pressure measurement of the pulmonary capillary pressure has given a value of about 7 millimeters of mercury. This is about almost correct, as the mean left arterial pressure is about 2 millimeters of mercury and mean pulmonary arterial pressure is only 15 millimeters of mercury, and thus the average falls around the 7 millimeter practically measured value. The length and the blood stay of the length of time the blood stays in pulmonary capillaries. When the cardiac output is normal, blood passes through the pulmonary capillaries in about 0.8 seconds. When the cardiac output increases, this can shorten to as little as 0.3 seconds. The shortening would be much greater were it not for the fact that additional capillaries, which normally have collapsed, open up to accommodate the increased blood flow. Thus, in only a fraction of a second, blood passing through the alveolar capillaries becomes oxygenated and loses its excess carbon dioxide. Next, we look into capillary exchange of fluid in the lungs and pulmonary interstitial fluid dynamics. The dynamics of fluid exchange across the lung capillary membranes are qualitatively the same for peripheral tissues. However, quantitatively, there are important differences. The first is that the pulmonary capillary pressure is low, about 7 millimeters of mercury, in comparison to the considerably higher functional capillary pressure in peripheral tissues of about 17 millimeters of mercury. The interstitial fluid pressure in the lung is slightly more negative than that in the peripheral subcutaneous tissue, which ranges from -5 to -8 millimeters of mercury. The pulmonary capillaries are relatively leaky to protein molecules, so that the colloid osmotic pressure of the pulmonary interstitial fluid is about 14 millimeters of mercury, in comparison to the less than half these values in peripheral tissues. The alveolar walls are extremely thin, and the alveolar epithelium covering the alveolar surfaces is so weak that it can be ruptured by any positive pressure in the interstitial pressure spaces greater than the alveolar air pressure, which is around greater than 0 millimeters of mercury, which allows dumping of fluid from the interstitial spaces into the alveoli and thus forces the alveolar wall to break. Next, we look into why it is necessary that we maintain a negative pulmonary interstitial pressure and the mechanism for keeping the alveoli dry. One of the most important problems in lung function is to understand why the alveoli do not normally fill with fluid. One's first inclination is to think that the alveolar epithelium is strong enough and continuous enough to keep the fluid from leaking out the interstitial spaces into the alveoli. However, if one remembers that the pulmonary capillaries and the pulmonary lymphatic system normally maintain a slight negative pressure in the interstitial spaces, it is clear that whenever extra fluid appears in the alveoli, it will simply be sucked mechanically into the lung interstitium through the small openings in the alveolar epithelial cells, and thus fluid does not accumulate in the alveoli themselves. In this particular diagram, we have a pressure. We see the various pressures that are present in the alveolar system. We see that the there is a net difference of about +1 millimeters of mercury that pushes the fluid into the alveoli, but it is sucked into the lymphatic system and does not let accumulation of fluid in the alveolus itself. Next, we look into the interrelations between the interstitial fluid pressure and the other pressure in the lung. The figure shows pulmonary capillary, pulmonary alveolus, and a lymphatic capillary draining the interstitial space between the blood capillary and the alveolus. That is this diagram right here. Note that the balance of forces at the blood capillary membrane as follows, which is given by the table right here. So first, we look into forces tending to cause movement of fluid outward from the capillaries into the pulmonary interstitium. These forces include the capillary pressure, the interstitial fluid colloid osmotic pressure, and the negative interstitial fluid pressure. which gives us individually gives us the total outward force on the blood present inside the capillary as 29 millimeters of mercury. The forces tending to cause absorption of fluid into the capillaries include the plasma colloid osmotic pressure, which is only 28 millimeters of mercury, and thus the mean filtration pressure in the pulmonary capillaries is plus 1 millimeters of mercury. Thus, the normal outward forces are slightly greater than the inward forces, providing a mean filtration pressure at the pulmonary capillary membrane, which is calculated as follows. Next, we look into the gas exchange between alveoli and pulmonary capillary. In this particular diagram, the outer layer right here is the capillary that is running from the pulmonary artery to the pulmonary vein, and the inner structure is obviously the alveolus. Here we can see that there is an alveolar membrane and a separate pulmonary capillary membrane as well. The gases can dissolve and diffuse between the lungs and circulatory system directly through the alveolar membrane and the capillary membrane, and we can see oxygen coming from the air into the blood vessel and being taken up by the RBCs, while CO2 is given out by the RBCs, and the CO2 gets accumulated in the alveoli and then cleared off as the lungs respire. Next, we'll look into the physical principles of gas exchange through the respiratory membrane. The first is the molecular basic for gas diffusion. All the gases in the respiratory physiology are simple molecules that are free to move among one another, which is the process called diffusion. For diffusion to occur, there must obviously be a source of energy. For any kind of movement to occur, there must obviously be a source of energy present. This particular energy, in case of diffusion, is provided by the kinetic motion of the molecules themselves. Except at absolute zero temperature, all molecules of all matter are continually undergoing motion. For free molecules that are not physically attracted to one another, this means linear movement at high velocity until they strike other molecules. Then they bounce away in new directions and continue striking other molecules away. In this way, the molecules move rapidly and randomly among one another. The net diffusion of a gas in one direction is essentially an effect of the concentration gradient of the gas. If a gas chamber or solution has a high concentration of a particular gas at one end of the chamber and a low concentration at the other end, as shown in this figure right here, net diffusion of gas will occur from the high concentration area toward the low concentration. The reason is obvious. There are far more molecules at the end A of the chamber to diffuse towards end B than there are molecules to diffuse in the opposite direction. Therefore, the rates of diffusion in each of the two directions are proportionately different, and we can see a net movement of molecules from the end A towards the end B. After looking at diffusion, we will look into the concept of partial pressures of individual gases. Pressure is directly proportional to the concentration of the gas molecules. In respiratory physiology, one deals with a mixture of gases, mainly oxygen, nitrogen, and carbon dioxide. The rate of diffusion of each of these gases is directly proportional to the pressure caused by that gas alone, which is called the partial pressure of that gas. Consider air, which has an approximate composition of 79% nitrogen and 21% oxygen. The total pressure of this mixture at sea level averages about 760 millimeters of mercury. Each gas contributes to the total pressure in direct proportion to its concentration. Therefore, 79% of 760 millimeters of mercury is caused by nitrogen, which comes about 600 millimeters of mercury, and 21% by oxygen, which comes about to 160 millimeters of mercury. Now, next we look into pressure of gases dissolved in water and tissues. Gases in water or in body tissues also exert pressure because the dissolved gas molecules are moving randomly and have kinetic energy. Further, when the gas dissolved in fluid encounters a surface, such as the membrane of a cell, it exerts its own partial pressure in the same way that a gas in a gas phase does. Thus, partial pressures are not only confined to the gaseous phase, but gases dissolved in tissues and water also have a partial pressure, and they can exert a pressure directly on the surface of a cell or the surface of a tissue. Next, we look into the factors that determine the partial pressure of a gas dissolved in a fluid. The partial pressure of a gas in a solution is determined not only by its concentration but also by the solubility coefficient of the gas. Some types of molecules, especially carbon dioxide, are physically or chemically attracted to the water molecules. When molecules are attracted, far more of them can be dissolved without building up excess partial pressure within the solution. Conversely, in case of those that are repelled, high partial pressure will develop with fewer dissolved molecules. These relations are expressed by the following formula, which is known as Henry's law, which is given as partial pressure of a particular gas is equal to the concentration of the dissolved gas upon the solubility coefficient of the gas. Next, we look into the factors that determine the partial pressure of a gas dissolved in a fluid. When partial pressure is expressed in atmosphere, where 1 atmosphere equals 760 millimeters of mercury, the concentration is expressed in volume of the gas dissolved in each volume of water. The solubility coefficient for various important respiratory gases at body temperature is given as follows. We can see if oxygen is taken as a solubility with a solubility coefficient of 1, carbon dioxide has a solubility coefficient of 20.3. That is, carbon dioxide is 20 times more soluble in water than oxygen. Therefore, the partial pressure of carbon dioxide for a given concentration is less than 1/20th that is exerted by oxygen as it is dissolved in a particular tissue or water. Next, we look into diffusion of gases between the gas phase in the alveoli and the dissolved phase in the pulmonary blood. The partial pressure of each gas in the alveolar respiratory gas mixture tends to force molecules of that gas into a solution in the blood of the alveolar capillaries. Conversely, the molecules of the same gas that are already dissolved in the blood are bouncing rapidly in the fluid of the blood. Some of these bouncing molecules escape back into the alveoli itself. The rate at which they escape is directly proportional to their partial pressure in the blood. The net diffusion is determined by the difference between the two partial pressures. If the partial pressure is greater in the gas phase in the alveoli, as is normally true for oxygen, then more molecules will dissolve or diffuse into the blood than in the other direction. Thus, we see a net movement of oxygen from the alveolus towards the blood. Alternately, if the partial pressure of the gas is greater in the dissolved state in the blood, which is normally true for carbon dioxide, the net diffusion will occur towards gas phase in the alveoli, and thus we will see a net diffusion of carbon dioxide from the blood into the alveoli, and thus carbon dioxide can be easily expired out of the body. Next, we will look into quantifying the net rate of diffusion. In addition to the pressure difference and solubility, several other factors affect gas diffusion, which include the solubility of the gas in the fluid, the cross-sectional area of the fluid, the distance through which the gas must diffuse, the molecular weight of the gas, and the temperature of the fluid. All of this can be combined into this particular formula, which gives us the diffusion coefficient as proportional to the the rate of diffusion as proportional to the pressure difference, the area or the cross-sectional area, the solubility of a particular fluid upon or the rate of diffusion is then inversely proportional to the distance the particular gas molecule has to travel and the square root of the molecule weight of the gas molecule. In the body, the last three factors the temperature remains reasonably constant and usually need not be considered. The greater the solubility of the gas, the greater the number of molecules available to diffuse for any given pressure difference. The greater the cross-sectional area of the diffusion pathway, the greater the total amount of molecules that diffuse. Conversely, the greater the distance the molecules must diffuse, the longer it will take the molecules to diffuse the entire distance. Finally, the greater the velocity of kinetic movement of the molecules, which is inversely proportional to the square root of molecular weight, the greater the rate of diffusion of the gas. In the particular given formula, D is the diffusion rate, del P is the partial pressure difference between the two ends of diffusion pathway, A is the cross-sectional area of the pathway, S is the solubility of the gas, D is the distance of diffusion, and MW is the molecular weight of the gas. The characteristics of the gas itself determine two factors of the formula, the solubility and the molecular weight. In the formula, only the solubility and the molecular weight will change as we change from one particular respiratory gas to another. Together, these two factors determine the diffusion coefficient of the gas, which is proportional to the solubility upon square root of molecular weight. Next, we look into the respiratory unit and the diffusion of gas through tissues. A schematic representation of the respiratory unit is given here. First, we look into diffusion of gases through these tissues. The gases of the respiratory importance are highly soluble in lipids, and consequently they are highly soluble in cell membranes. Because of this, the major limitation to the movement of gases in tissues is the rate at which the gases can diffuse to tissue water instead through the cell membranes. Therefore, diffusion of gases through the tissues, including through the respiratory membrane, is almost equal to the diffusion of gases in water. Next, we look into the respiratory unit. The respiratory unit is composed of a respiratory bronchiole, alveolar ducts, the atria, and the alveoli. There are about 300 million alveoli in the two lungs, and each alveolus has an average diameter of about 0.2 millimeters. The alveolar walls are extremely thin, and between the alveoli is an almost solid network of interconnecting capillaries, which is shown in the figure in the right. Gas exchange between the alveolar air and the pulmonary blood occurs through the membranes of all the terminal portions of the lung, which is collectively known as the respiratory membrane or the pulmonary membrane. So this is the particular respiratory unit that is shown right here. Next, we look into the surface of the cross-sectional view of the alveolar walls. So this is a surface view of the alveolar walls, and you can see so many pores are present along the alveolus. And this is a cross-sectional representation where we can see the alveolus present here, which is divided from one another via the capillaries, and the interstitial spaces, the lymphatic vessels are also shown. And these are the major veins and the arteries that are passing through the region which are connected to the alveolus via the various capillaries. Here we have an ultrastructure of the alveolar respiratory membrane, and we can see a blown up of how the diffusion occurs from the RBCs to the alveolus. You can see right here that there are the number of membranes that are present between the alveolus on the left and the blood capillary on the right, which include the alveolar epithelium, the epithelial basement membrane, the capillary basement membrane, and the capillary endothelium, and via which the diffusion has to occur. There is also the fluid and the surfactant layer that we have already discussed present on the alveolar epithelial side, and the interstitial space between the alveolar membrane and the capillary membrane. We'll look into more into the respiratory membrane in subsequent slides. So the figure here shows the ultrastructure of the respiratory membrane drawn in cross section on the left and an RBC cell on the right. Note that the following different layers of the respiratory membrane that are present. First is a layer of fluid lining the alveolus and containing the surfactant that we have already discussed before that reduces the surface tension of the alveolar fluid. Next is the alveolar epithelium composed of thin epithelial cells followed by the epithelial basement membrane. Next is a thin interstitial space between the alveolar epithelium and the capillary membrane. Next is the capillary basement membrane itself that in many places fuses with the alveolar epithelial basement membrane. And finally, we have the capillary endothelial membrane. Despite the large number of layers, the overall thickness of the respiratory membrane averages about 0.6 micrometers. The total surface area of the respiratory membrane is about 70 square meters in the normal adult human male. The total quantity of blood in the capillaries of the lungs at any given instant is 60 to 140 milliliters. This amount of blood spread over the entire surface of the 70 square meter explains why the whole process of respiratory exchange of oxygen and carbon dioxide is so rapid. The average diameter of the pulmonary capillaries is only about 5 microns, which means that the red blood cells must squeeze through them. The red blood cell membrane usually touches the capillary wall, which greatly increases the rapidity of the diffusion. The final topic that we'll look through here in these slides are the factors which affect the rate of gas diffusion through the respiratory membrane. The first factor that we will look into is the thickness of the respiratory membrane. The thickness of the respiratory membrane occasionally increases. For instance, as a result of edema fluid in the interstitial space of the membrane and in the alveoli, so that the respiratory gases must then diffuse not only through the membrane but also through this edema fluid. Because the rate of diffusion through the membrane is inversely proportional to the thickness of the membrane, any factor that increases the thickness to more than 2 to 3 times normal can interfere significantly with the normal respiratory exchange of gases. Next factor that we'll look into is the diffusion coefficient. The diffusion coefficient for transfer of each gas through the respiratory membrane depends on the gas's solubility in the membrane and inversely on the square root of the gas's molecular weight. As we have already seen in the previous formula, the diffusion coefficient is directly proportional to the solubility of the particular gas divided by the root over of molecular weight. The rate of diffusion in the respiratory membrane is almost exactly the same as that in water, for the reason that we have already seen. Therefore, for a given pressure difference, carbon dioxide diffuses about 20 times as rapidly as oxygen. Similarly, oxygen diffuses about twice as rapidly as nitrogen. Next, we look into the other two factors that are involved in the rate of determining the rate of gas diffusion, which include first the surface area of the membrane. The rate of gas diffusion is directly proportional to the surface area via which diffusion occurs. The surface area of the respiratory membrane can be greatly decreased by many conditions. For instance, removal of the entire lung decreases the total surface area of the to one half now, which is obvious that if we remove one of our two lungs, the entire surface area will decrease to half. And this in turn affects the surface area of the respiratory membrane that is available for diffusion and thus reduces the rate of overall diffusion. Also, in case of emphysema, many of the alveoli coalesce with dissolution of many alveolar walls, which again decreases the overall surface area available for diffusion. Finally, we look into the partial pressure difference. The pressure difference across the respiratory membrane is the difference between the partial pressures of the gas in the alveoli and the partial pressure of the gas in the pulmonary capillary blood. The partial pressure represents a measure of the total number of molecules of a particular gas striking a unit area of the alveolar surface of the membrane in unit time, and the pressure of the gas in the blood represents the number of molecules that are attempting to escape from the blood in the opposite direction. Therefore, the difference between these two pressures is the measure of the net tendency of the gas molecules to move through the membrane. Thus, the difference between the partial pressures between the gaseous phase and the dissolved phase present in blood determines the rate at which diffusion will occur and also the direction of the diffusion. As we have seen, carbon dioxide is, as we have seen with respect to carbon dioxide and oxygen is more readily dissolved in blood, while carbon dioxide is more readily expired from the blood and travels to the alveolar air sacs, and thus is expired off easily by the lungs in the process of expiration. Thus, in this particular context, we are able to appreciate how partial pressure difference between the gaseous and the dissolved phase help in determining the rate of diffusion as well as the direction of diffusion. This was all we had for today. The references to these particular slides are the chapters 37, 38, and 39 from the textbook of medical physiology by Arthur C. Guyton, the 11th edition. I thank you for listening to the video.